Welcome, everybody. My name is Benjamin Allen. I am a technician here at Oak Ridge National Laboratory on the DOE Systems Biology Knowledge Base, aka KBase project. I'm joined by my colleagues Ellen Dow as well as Zach Crockett, who are going to be helping with the webinar today to answer questions as we're going through all of the material. And yeah, I hope by the end of this, you'll get a good sense of what KBase is, how as a platform and as a scientific project, as well as what you can do with the platform and the tools and the data inside of the platform to allow you to drive your research and analysis and access some of the power that's available in the platform to you all. So I'm going to go through a couple of quick slides here just to give some context for what the KBase project is. And again, you can feel free to reach out to me with any questions you have or any thoughts or inquiries about using the platform, or if you have anything you want to touch base with us on, you can reach out to me at allenbh@ornl.gov, and we have some other ways you can contact our team as well if you have tickets or troubleshooting issues as you're using the apps and tools on the platform, or if you have just general inquiries about how to integrate KBase into your research. I'm just gonna, all right. So what is KBase? We are a community-driven research platform for systems biology. So we really rely on a wide-ranging user base of scientists and researchers across the world who are utilizing the tools and data in our platform to drive their research. So as of this recording, we have about 40,000 accounts, and the majority of those accounts are active users. So we're always excited to introduce the platform to new users and new researchers to try to onboard them into using KBase to drive their research. So we really have a couple of goals with the platform. One is to make an accessible platform that regardless of how skilled people are in the computational science side of bioinformatics, biologists and other researchers can access the tools that they need to drive their research in a easy to use and accessible environment. So to do that, we number one, we try to integrate tools and databases into KBase that are commonly used or represent kind of the best in class as far as different categories of tools. We pull those into the platform and try to keep them up to date with their current state so that you are able to access these tools and run them on your own data so that you can not just analyze your own data, but you can also analyze your data in the context of public data. So in addition to the tools we bring in some of the relevant reference databases that are especially useful for doing comparative genomics and other kind of comparative analyses. We also try to support researchers working together. So we have a couple of features that enable collaboration that I'll demonstrate later on in the webinar. And additionally, we want you to publish the results and data that you have generated on the platform. So we have some publishing features as well that we are excited to share with you all, and certainly excited when we see people integrating these into their publications. So KBase is funded by the Department of Energy Office of Science, and specifically the Biological and Environmental Research program within the Office of Science. So BER within DOE funds a lot of different projects. You can see some of them here. Many of you may be familiar with the Joint Genome Institute, which is the premier sequencing facility that Department of Energy sponsors, alongside the National Microbiome Data Collaborative, which is a project that is trying to help encourage people to standardize their samples and collection data, as well as tie that to their sequencing projects and other data that they're generating from environmental samples. And so our kind of role within this ecosystem is to build a platform that enables the analysis and visualization, as well as, again, sharing and publishing of the data that people are generating either from these institutes, or perhaps you have a sequencing center at your university that you are utilizing. So again, our function is to create a online web-based platform that integrates these different types of data and analyses and serves them up to the research community for biology. So some of the ways that we do this, as I already mentioned, is integrating data from public databases, as well as some DOE-specific projects that have their own curated data sets. We're pulling in tools from the open source bioinformatics community. And importantly, we're connecting those tools to Department of Energy supercomputing and high performance computing platforms at Argonne National Laboratory and Lawrence Berkeley Laboratory, such that you are accessing some of the high-powered computing required to drive some of these tools, especially on large data sets. As sequencing gets cheaper and as it increases in accuracy, and as the number of replicates and number of samples increases, we want to make sure that we can adequately provide the amount of computing power required to process some of those data. So yeah, we hear often from our users that they may have tried to build their own supercomputing cluster, or they may have utilized the one that their university offers, or maybe they don't have one at their university, and they know that they need to have some high performance computing arrays to do some of the computationally intensive processes like metagenome assembly, for instance. So this allows us to power these tools using the Department of Energy supercomputing infrastructure. And importantly, we don't just offer a there are some other bioinformatics web platforms and services that don't offer you a lot of parameterization or customization. So we really encourage the development of custom analytical pipelines that allow you to combine multiple modes of analyses to drive your research. And again, we want you to share your research not just with your collaborators, but with the broader scientific community through your publications. So we both enable you to work with other researchers in your lab or between institutions, and then share that with other people who may be interested in your research. So the primary working environment of KBase is built around what we call narratives. So narratives are digital notebooks that contain all of the data, the tools, and any commentary or scripting that you've done alongside of your analysis. So if this appears familiar, this is built around what's called Jupyter notebooks. So some of you may have used Jupyter notebooks for your data analysis, or using it for different kinds of computational science practices. So they're, of course, increasingly popular across all scientific disciplines, but we are specifically nesting this within KBase to be useful to biologists as well as bioinformaticians who are looking to um, create a repository and a reproducible environment that contains all of their data and analyses. So what narratives really allow us to do is not just create a kind of graphical user interface where researchers can point and click and configure parameters and things like that in a pretty straightforward way, but also create these research products that when you're sharing them in your publications or sharing them um, with other researchers, they're able to look and see exactly how you set the parameters on different tools or the versioning of different tools and data that went into your analysis. So this is important as far as helping with reproducibility in science. As I said, we enable a variety of customizable analytical pipelines. I'll show you how these are broken down in the narrative and how you can kind of select from different flavors of tools. But you can just see here what we offer you is a way to not just run some singular pipelines, but to connect those things together. So for instance, if you have an isolate organism that you have recently sequenced and you are looking to process, assemble, and annotate that, you don't have to just stop there. You can move into other pipelines such as metabolic modeling or comparative genomics, where you can compare your novel organism with other organisms, or get a sense of its metabolic potential represented within its genome. So again, we really hope to offer something where you have a lot of flexibility and customization as far as how you are building out your workflow. And additionally, we offer a variety of tools in each of these categories, so you can also compare how different tools, the outputs and results of running different tools on your data, how different tools may impact your findings. So there's a lot of customization possible in the platform. And so we have over 200 apps that fall into a couple of different categories. So you can just see that there's a wide array of what you can do on the system. But we also have a software development kit that enables computational biologists or computer scientists who may be developing new bioinformatics tools to integrate them into the platform such that they're serving them up to all of you and the broader research community. So, instead of, putting them just into a GitHub and hoping people will come and find them and figure out how to install the tools and all of the dependencies and all of these things, the platform enables those developers to situate their tools in a way that makes them easier to use and more accessible, in addition to driving them with our high performance computing power. I'll demonstrate data upload and import more directly, but just to say we enable both a kind of easy way to drag and drop data into narratives. But if you have, gigantic data sets with, hundreds of files, there are some ways, especially using Globus, that you can more straightforwardly transfer data into KBase for analysis. So, we often find that, ease of data import is a very key way to build trust with the platform, because if people can get their data in easily and start to work on it quickly, they're going to continue to come and use the platform for their analysis. So we're very interested, of course, in making it easy for you to import your data if you are exploring the platform and you find that we don't have certain data types, or you wish, things operated a little differently, please let us know, because we're very interested in making it seamless and easy for you to get your data into KBase. Um, and again, public data integration is crucial for this idea of analyzing your private data in the context of public data, because, it's very difficult for someone to, for instance, pull all of the available genomes from RefSeq onto their own computer. To build out, some, all versus one versus all type analysis. So we have built into the platform a couple of key public databases, including the RefSeq collection from NCBI here, run by the NIH in the United States, as well as Joint Genome Institute genomes from Phytosome and Microcosm for plants and fungal genomes, respectively. So we're also trying to be kind of cross-domain as far as what kinds of organisms and communities we're representing in the system. So if you have additional suggestions for public resources you'd like us to integrate, we're also interested in hearing about that. We are also working to pull in viral and microbiome data from JGI, as well as working with NMDC to harmonize the way we represent samples, sampling projects from different environments that are tied to sequencing projects. So we're looking to integrate a variety of data from public projects as well. As I mentioned, you can work with other users. So within a narrative, you have the ability to share your work with and share access to the narrative with other collaborators or KBase users. So, we're probably all very familiar these days with working in like Google Docs, where you can have multiple people operating on a single document. While you can't necessarily have, several people in the same narrative, you can pass these things back and forth. And you can control what people who have permission to access your narrative, what level of permission they have, if it's view only, or they can edit and save, or they can edit, save, and share. By default, all narratives are private, so all of your data and your analysis is kept private unless you choose to make it public. But you can also just make a narrative open to the public, meaning that anyone with a KBase account can come and have view-only access to your narrative. Another way you can collaborate with people is through organizations. So you can build a organization that may have a defined list of users, perhaps from your lab or from your department at your university, such that you are maybe collaborating across a series of narratives, and you want to make sure that everyone in the organization can have access to those. So there's ways you can kind of more easily maintain a controlled list of users that you want to share with as people enter and exit, a lab or a research project, such that you can maintain access to the work that they've done on the platform. And another way that we're serving up public data is with this new feature called collections. So collections is a new data environment that we just recently released earlier this year. And you can see that we've got a couple of projects that we have worked with as the initial set of collections that we've released. And so what a collection allows you to do is, if you have if a project has a list of a set of genomes that they have been working on that they want to enable other researchers to access, or they kind of want to serve them up to the larger community. We can build a collection of those genomes, and this allows other KBase users to come and access the curated set of genomes that a project has made. And it also allows them to do some searching and finding based off of either sequence similarity or taxonomic similarity, of their private data against these collections. So you can see, for instance, there's this Genome Resolved Open Watersheds project that has integrated their genomes into the platform. And so these are all isolates drawn from different kinds of river environments. And so if you are studying river ecology, or you have an isolate from that kind of environment, you can search for that against the GROW collection and see if there are any hits or similar genomes in that collection. And you can see too, there's additional ways that you can kind of explore the data in these collections, based off of geography, as well as, biological characteristics. And you can get a sense of the quality of the genomes. Some of these are isolates, some of these are high quality mags, but you can kind of explore the data from a variety of ways. And so, yeah, with publishing, we have what we call static narratives. I think we're going to move to calling these fair narratives, as in findable, accessible, interoperable, and reusable, because we want, as you're going through an analysis on the platform, right, you're operating with the data and the tools that you have at a specific time. But as we know, data is always changing, tools are always changing. So what a static narrative enables you to do is create a snapshot of your work at a specific point in time. So at the point of publishing, for instance, you have a specific snapshot of your narrative that you can release to the public, but you can still maintain and develop your work in a narrative on the platform without affecting the narrative that you've shared in your publication. So maybe new data is released or new tools are released. You don't have to create a fork necessarily. You've got this snapshot that's a persistent identifier that you can link in your paper that will take external people to the exact version that you had for your publication. And you can also register a DOI for your static narrative. Again, if you may have results that you're wishing to share across multiple papers, or you may be trying to build your own kind of collection, and so you want to have DOIs for your static narratives in addition to the DOIs for the paper that you've published. So you have some additional kind of research objects that you can generate. And so that can be not just useful for publishing, but for your CV, or for your tenure package. You can have GOIs linked to multiple narratives and data products associated with what you produced on the platform. And so we've partnered with ASM Microbiology Resource Announcements to build a collection of MRA announcements that are linked to these static narratives with DOIs as well. So just additional layers of kind of promoting the work that you've generated on the platform. And this is just an example that we have from some users that have generated some mags from the Amazonian forest. We also allow you to integrate amplicon and environmental sampling data to your your projects within KBase. We'll be doing some more in-depth training on this later this year and early next year. We have a few webinars about how to use these features right now. But just know that if you have, for instance, amplicon data that you're looking to link to genomes that maybe are your own genomes or in these public repositories, we are building out a service that allows you to host your experimental data and sample metadata and analyze that in the context of this public data as well. So the kind of overall vision of what we're trying to do with the platform, in addition to building this kind of community-focused and accessible platform for bioinformatics, our scientific mandate from DOE BER is to really harden the computational representation of a variety of systems and scales for Earth systems and biological analysis. So you can kind of see our understanding of what we are trying to link together between various data types and various research objects, I guess, from everything from genomes and organisms to the environmental context that they're in and the geochemistry, as well as information about their geolocation and when the sampling took place. So that's the kind of grand challenge of the KBase project, is to design and link all of these data types together in ways that are meaningful for researchers and usable within the platform. And another way that we're doing that is with something we're actively building, what we call a data transfer service that will allow us to, just as I showed you at the beginning, our kind of linkage with other Department of Energy BER funded projects. We want to build a portal and a transfer service that allows us to integrate data from all of these other projects into the platform and then serve that up to you all as a research community, so that you're not just accessing, for instance, RefSeq genomes, but you can also access projects on NMDC or metabolomics data from the Environmental Molecular Sequencing Laboratory, and do that directly within the platform. If any of you are teaching undergraduates or early graduate classes and you are looking for curricular materials to integrate bioinformatics into your teaching, we have a educators program led by Ellen Dow, who is on the webinar today, and she has built a community of about 200 or so educators that are using KBase in their classroom to build and drive student projects that demonstrate bioinformatics to these students and get them excited about this aspect of biological science. So if you are an educator and you'd like to join that program, you can follow this link on our main page to the educators program, or you can reach out to Ellen and she will definitely reach out to you and integrate you into this educators project. Finally, this is just some statistics about the number of users and publications that we are generating on a year-to-year basis. This is a little older, but we're actually just at the end of the quarter, so we're about to start to put together the latest version of this. But we've been happy to see that just really in the past 5 years or so, a large number of users have had success using KBase in their research, and we're seeing the number of publications and the sophistication of those publications, in terms of how many different kinds of analytical workflows that are represented in their research, increasing over the years. So we get about 125, 130 or so, proper methodological citations every year. And I think this year we're going to beat out all of our previous years as far as the number of users who are successfully publishing with us. And so this is our team. We're a relatively small team given the scale and complexity of the platform, but we're spread across 4 different national laboratories, with the majority of the team based at Berkeley Lab, as well as the many of us at Argonne, Oak Ridge, and Brookhaven. So we are kind of unique as far as Department of Energy projects go in terms of being a multi-laboratory initiative. And so it's been great working with all our colleagues across the various labs to build the platform. And this is just a link to our project paper. This is linked out in the questions and answers document, but if you want to see kind of the design vision and principles of how we built the platform, you can visit this publication from Nature Biotechnology from 2018. All right, so I am going to go ahead and start with the proper demonstration of the platform here. To get to KBase, you go to kbase.us here, and you'll see just, some basic information similar to what I've already shown you on our homepage. The only other things I really want to point out here that are going to be very useful to you all, just starting at the right-hand side of the toolbar at the top. One is our support and help board. So as you're using the platform, you may run into bugs or issues, or just have general questions about how to do something. So I would encourage you to take a look at our help board. There's there's also good examples of people that may have had similar questions to you. So you may be able to identify someone else has already had the same kind of issue as you. So I just recommend people taking a look at the KBase help board, because it's got, it's very likely that if you're having an issue, someone else has run into that issue as well. And as you can see, my colleague Zach Crockett helps lead our help board effort. And so if you have an issue, Zach will be very quick in responding and helping you out. Other things to check out are our news section. So whenever we have new features, or we have, for instance, examples of our users who are having success with the platform, we try to put these up on our news page so that you all can see what we're doing there. I'm just going to go ahead and sign in to the platform. If you don't have an account already, you can go to this sign up button here and create an account relatively quickly. As you'll see when I'm signing in here, we have a couple of different authentication methods that allow you to sign in. So I'm sure all of us have a Google account these days, so you can just sign in directly with your Google account. What we are encouraging you all to do is if you don't already have an ORCID account, you can create one and sign in with that. And this is sort of our preferred way for people to sign in, just because it allows, as we're building out some of these features like static narratives, we're exploring ways that we can create additional object and data identifiers that directly tie to your ORCID account. So consider using your ORCID identity for sign-in. We also have the Globus as well. This is something that specifically those of us with Department of Energy, that the Department of Energy has encouraged us to use. So this is why I have a ORCID or a Globus account here. So what you'll see as soon as you log into the platform is this narrative navigator section. So as I mentioned, narratives are the primary working environment of KBase. So just about everything you do in the platform is contained within a narrative. So the first page when you log in just shows you all of the narratives that you own, and gives you some information about what's in each of these narratives so that you can find the right one. You can do searching, you can do filtering to find the right narrative. If you don't have anything on the platform yet, it'll of course just be blank when you start, but you can just see that there's ways you can see what's in your narratives without having to actually open them up, which can be useful when you're working on a large number of projects. You can also see any narratives that are shared with you. So if you are collaborating with different researchers, you can use the Shared With Me tab to identify those narratives that have been shared with you. We also have tutorials. So we've tried to create tutorials for all of kind of like the main analytical pipelines that are possible on the platform, and work to keep these updated as, again, tools and data evolve. Um, and we also try to do webinars on each of these different kinds of workflows. So we recently did one on the viral genomic tools in KBase. So if you go to our YouTube, you can see the recording of that tutorial and training. But you'll see there's trainings for metabolic modeling, extracting mags from environmental samples, drafting isolates, and doing comparative genomics and feature analysis in those isolates, and so on. So we try to build out tutorials that generally we think users will be interested in. If you have suggestions or anything you don't see here, we're happy to hear that from you if you want to let us know what you think we should build next. And finally, you can see public narratives. So again, these are narratives where the user has decided to allow them to be public and accessible by anybody. So some of these are linked in publications. Some of them, maybe they just similar to enabling access to anyone with a link in a Google Doc. They just wanted to create a single accessible link for anyone that's a KBase user. So you'll see this is just ways you can kind of manage narratives and access them, and as well as learn about different pipelines on the system. What I'm going to do here is go ahead and create a new narrative. So in the top right corner, you'll see this new narrative button. And when I click that, it will start to spin up the narrative here, and we can begin to demonstrate some of the first steps typically. If you're ever on this screen and it gets stuck, that typically will only happen if maybe you have a weak internet connection, or there is tons and tons of data in a single narrative. Your options are to be patient, or you can do a hard refresh, and that will usually kick it over and open the narrative. So this is a blank narrative. So we have no data or tools or anything in there, just this welcome cell. So if you're in the narrative and you want to access documentation, you can of course click any of these links in this welcome cell. This is just showing that I'm logged into WordPress right now, but so you won't see that. But so let's go ahead and walk through the different aspects of the narrative. So the first thing I'm going to do here is provide a title. So I just click the title, which is currently untitled, up in the top left corner. And I will just type in a name here. And this will save a copy of my narrative here. So other items on the toolbar, you'll see on the right-hand side the save button. So you can manually save, and KBase will also automatically save anytime you add data or run an app. So there is an autosave, but still it can be useful to manually save occasionally. In addition to that, there's the share button, which allows you to do sharing with other colleagues and create static narratives. And there's some additional items. So for instance, if you're in the narrative and your internet connection drops for whatever reason, it will show that you're not connected. And so you can use this kernel button to restart or reconnect. So sometimes that happens if that's necessary if you lose access due to an internet connection issue. But let's go ahead and let's add some data so I can show you how to get your data into the platform. So on the left-hand side here, you'll see this data panel, and it says this narrative has no data yet. So I'll click this Add Data button, and this opens a slide out that has some various categories as well. By default, it just shows my data. So this is data I've already uploaded and imported into other narratives. So maybe I want to find some other narrative where I have some data and I want to pull it into a new narrative, or I have a specific type of data that I'm looking for. This will just show you any data that you have already imported into the narrative. Similar to the navigator, I can see data that's been shared with me. I can see public data. So as I mentioned in the intro, we have these various public repositories that we have integrated into the platform. So maybe I want to, find some organism that's in RefSeq. I can search for various terms and I can add those genomes into my narrative very straightforwardly by, I can just click this add button and you'll see it appears in my data panel and is ready for analysis or other processing. I'm just going to hop over to this import tab since that's what we're really looking at here. And I'm going to open on my screen some example data just to show you how you can kind of import some multiple types of data. So I've got some reads, I've got assemblies, and I've got genomes. I'm just going to drag and drop these into this drag and drop section, and you'll see that those begin to import. Obviously, this 1.9 gigabyte reads is going to take a little while, but you'll see that the genome and the assembly uploaded pretty quickly. So there's kind of a two-stage process with getting your data into the platform. There's the upload part, which is what I'm showing you right now, and then there's the import. So you upload your data into KBase, and then you import that data into your specific narrative. So when you upload, it goes into this staging area. So the reason it's set up like that is because maybe you've got a lot of data that you're trying to upload but you want to distribute it across different narratives. So it goes into your staging area first, and then you can pull that into your specific narratives. One thing to note about this drag and drop interface is that just with the limitations of current browsers, Chrome, Firefox, Safari, we do not recommend trying to import anything over 20 gigabytes. Generally speaking, there's a limitation with how browsers can handle data of a size larger than that, and it cuts out around between 15 and 20 gigabytes. So if you have any data that is larger than that, you should use Globus. And so we have documentation on our docs site docs.kbase.us, where you can understand how to set up a Globus import endpoint to import your data. You can also import with a URL. Maybe you have data that's on an FTP server on your university or somewhere else on the web, like a Dropbox or Google Drive. You can use this upload with URL, um, and that will allow you to just drop the link to the data that you want to pull into your narrative. So that's a couple different ways you can get your data into KBase. So I'm just going to go ahead and import this data now. So you can see my genome here is at the top of my list in my staging area. It has automatically selected what type of data this is. This is a GenBank flat file, so it automatically determined that this is a GenBank genome. In the suggested types, you can see other types of data that you can import. So this next one, this assembly, this one says select a type because it couldn't determine exactly what it was. So in this case, this is a FASTA assembly, but you'll see, of course, when you are, there's people have different preferences for how their file types for their genomes are organized, and very popular way is to have your GenBank flat file alongside your FASTA assembly to compose your genome. So that's two files. So you'll have to select this for both your FASTA and your genome flat file if you have that specific type. But in this instance, we're just demoing the assembly and the genome. So I've selected the two files that I want to import here with this radio box selection. If I wanted to download any data or delete them from my staging area, I can do that as well, but we just want to import. So I click that import button, and then it opens up the last stage of the import process. So this has opened up what we call a cell in your narrative that allows me to specify some additional metadata for these files. So for instance, for this GenBank genome, you can see on the left-hand side of this importer that it's organized these files that I want to import into different types. So I see my assembly and I see my genome. The path is automatically selected. I just can, add additional metadata. Like for instance, this is a finished isolate, but if it was a MAG, a SAG, virus, plasmid, I can select that as well. This did come from RefSeq, and then if it has some hit as far as NCBI taxonomy goes, I can also specify that as well. So this one has a NCBI taxonomy ID. If you have a uncultivated or unspecified or unknown, there are also NCBI taxonomy IDs for that that you can select. But this just allows us to make sure that we are properly labeling any of the data that comes into the platform. And similarly, with the assembly, I see I've got some different parameters, and these are fine for this. So when I have set everything into the importer, I can, you'll see it says ready to run here. And I see this run button, so I will click that, and it begins to launch the job. So what I just did there, launching the job in this narrative cell, is very common practice in KBase. So as you add apps and data and you are running different tools, you do this process a lot, which is filling out the parameters, setting the inputs and outputs, and then you click run, and this is how you start jobs on the platform. So you can see that this is running in the background. Each job here, if I click where it says running, I then get access to this log here, which tells me, gives me the line by line of what's happening as this job is running. So, and very quickly they're finished, and you can see in my data panel now I have all of the data that I wished to upload. So it has closed out this job. This job will close out shortly. If I want, if say I'm importing 10 different objects and, 8 of them were successful, but some of them failed or some of them were canceled, I can, click this cancel retry all button, and I can, cancel, either running jobs or queued jobs, or I can retry those ones that I've canceled. So that can be useful occasionally. So, okay, we've got some data into our narrative. One thing I just want to do quickly is I want to shrink this welcome cell. So if I go to the top right of this cell, I'll see that there's some controls for this, including this collapse cell. So yeah, as you're working in the narrative, you can kind of keep things clean by managing your narrative cells, and you can move them up and down, or you can collapse them. So this just helps you kind of arrange things. So now I want to run an app on one of these tools. So I see in the bottom left-hand side, I've got the apps panel. I've got all these different categories. So if I open up a category, I can, like genome annotation there, what I'll see is a list of all of the apps under that category. And let's see, so I can explore all of these different categories here. I'm going to, I can also search for apps. So I'm going to search for a one that we have that builds out a phylogenetic tree. So it's just typed in this string insert here, and I found this insert genome into species tree app. When I click that, it will load it into my narrative here. And again, it's pretty straightforward to run all of these tools. You just set the input data. So in this instance, I'm just going to grab the genome that I imported. I can set the number of, in this instance, the number of public genomes that it's going to compare my input genome to build the species tree. And I can just set these parameters, and we'll just give these a name. And once I've set all the parameters, it will now show me that this app is ready to run. If I didn't set a parameter that's required, I cannot click this button. You'll see it's grayed out. And then there's this little red flag on the right-hand side of the parameter that's missing. So then I know I have to provide something. So if you're ever getting the grayed out run, it just means you haven't provided all of the parameters required to run the app. So I click run, and again, this job has now launched, and it will keep running for another 5 minutes or so in the background. So again, very quickly here, just in like 15 minutes, I've gone from importing data into the narrative and running the job. So it can be a very quick process overall to get jobs running on the platform. So I just want to show a couple of things while this is running now. For one, there are ways you can select apps by specific type. So, right, I have a genome here. Each data type I have uploaded has a specific category. So this is an assembly, this is a genome. If I click this 3 buttons here, I'll see some ways I can manage my data in my narrative. So I can delete data, I can rename it, I can download data off of the platform onto my own hard drive. So you can do all of this just by clicking this little ellipses next to the data name. And I can even just see which apps will take this type as input. So if I click this show apps with this as input, I'll see that it has added now to my apps panel a filter that only is showing apps that take a genome as a type. So for instance, I see these different metabolic modeling tools. And if I change that to assembly here, I'll see that it changes that list even further. So there's just different ways you can kind of explore apps on the system. Another thing I want to show you all is that what you're seeing here in the apps panel is released apps. So this little R button here allows me to toggle between release and beta versions. So if I click this button, what it will then show me are apps that have been released in beta. So they may not be kind of the alpha status fully tested and documented, but you can kind of see what's coming down the pipe as far as new tools on the platform. And so you'll just see that there's quite a few additional apps, and especially a large number of uncategorized apps that are all tools that are in development. So you can help us test these apps. There's a link in the Q&A document to sign up to be a beta tester. But of course, if you just want to explore these tools and run them on your own, that is more than welcome. And feel free to report whatever is if you run into any bugs, you can report those on our help desk. So one other thing I just want to show in the narrative quickly is if you see the bottom right-hand side of my screen, I've got two additional buttons. One is for adding a markdown cell, and one is for adding a code cell. So as I mentioned, this is built around the Jupyter notebooks, and so this allows us to run scripts directly in the narrative, or I can just add, context like, typing some text in here. And so I can, if I want to organize the different stages of an analysis and so on, or create hyperlinks, drop in images, put in different color text, link to other narratives, you can do that with the markdown cells. And additionally, if you search in our public narratives, what you'll find are some code cell tutorials about how to use the code cell. So this job has finished, and unfortunately, it's giving me this what is a known error right now. If you look at the help desk, a couple of people have been running into this error, and it's just related to our cookies right now. But this job has finished. Oops. If I can download the tree here and show it, but generally speaking, these apps will produce either a report or additional objects. So in this instance, I can just click the tree object here and load it in, and you are able to see the tree that it's generated for my genome. Anyway, this is just a known bug that came up after our maintenance window last week, so we're actively working to resolve this. But normally you would just see this tree inside of this cell. So you can fix this by clearing your cookies. I thought that logging out and logging back in would do that, but apparently not. So anyway, but we're working on resolving that issue right now. I know we're just a little over time, so I just want to hop back to the navigator. So this is what I showed you here is all of the basic aspects of using the narrative. So how to import data, how to add tools and how to configure tools and run them. Just want to hop back to the navigator here and show you a couple of other things I mentioned in the slides at the beginning. So you'll see you can access all of these different features on the left-hand side of the navigator. So the organizations, for instance, orgs will show me all of the various orgs that have been created by different groups. I can filter by all orgs. And so you see we've got 234 organizations. So you can see that people are using these for their classrooms so that they can build curriculum and exercises for their students, as well as there's some other orgs for different projects. If you want to see the various apps on the platform, you can go to the catalog here, and this will show you the app catalog page where you can just get a different view of all of the tools that are available. And you can search for them, or just if you find something you're interested in, like check-in, this is a very popular tool. You can click the tool name and it will take you to an information page that shows you documentation about this app. You can get a sense of how many people have run the tool, how successful it has been, and the average run time. In addition to, you can actually find links to the GitHub repo that shows how the tool has been implemented in KBase, so you can check the KBase version of a specific app, as well as look at information about publications or other links that are relevant for an app. The other thing that's useful is this jobs panel. So as you are, if you are running lots of jobs and multiple narratives, the jobs panel can be useful for monitoring various jobs. So you can see which narrative, which app, when it was submitted, how long it ran, what the status is, and you can observe the logs. So this is just another way you can, if you're running a huge number of jobs, you can manage them through this interface outside of the narrative, where you have a little bit more screen real estate. Account, of course, allows you to change different information about your account, and especially if you have multiple, so maybe you have an ORCID identifier, a Globus account, a Google, we often see people will create multiple accounts. You can link your multiple accounts through this interface. So I just like to mention that because we occasionally get tickets where somebody can't find their old account, but they made a new one, and so it can be useful to link accounts as well. And then I mentioned this in the slides, but we also have this collections feature that we're testing. So I can go open up one of these collections here, like plant microbe interfaces, which is from a group here at Oak Ridge. And I can look at all of the genomes within this collection in case, there are organisms of interest, or I'm trying to look at things from a similar environment. And if you have a project or a collection of curated genomes that you would like to integrate into KBase, let us know and we can help you build out one of these collections. Again, this is a brand new feature, relatively speaking. So, we're really trying to see how users may use this, or especially if you, again, are generating data, how you might like to integrate that into the platform, and so on. So, okay, I think that's a lot of information, of course. So just to, again, we'll be posting this webinar on our YouTube page. So I'll quickly show you our YouTube page where if you want to see additional trainings and webinars, at DOE KBase, you can see that we post up all of the various trainings and webinars that we do. So there's a variety of stuff up there. We'll be announcing additional workshops soon. So for instance, we will be showcasing some of our long reads specific apps. So if you have Oxford Nanopore or PacBio data, on October 23rd, we will have a webinar focused on using those tools. And in addition to that, there is our documentation page at docs.kbase.us. And you can just we've broken this down into various sections similar to how I just showed you all about how to do different functions on the platform. So you can consult the documentation as well as you can reach out to us directly at engage@kbase.us, or you can reach out to me directly, alanbh@ornl.gov, and I'm happy to work with you in addition to using our help board to report any bugs or issues or make feature requests. So I'm just going to take a look at our Q&A here and see if there's any unanswered questions. Great, looks like you all had a lot of questions, and thanks to Ellen and Zach for answering these. Yes, so there is you can create a gene tree. Are there any, let me open up my participants list here. Do we have any additional verbal questions or any other inquiries that people have today? See in the chat. Great. Yes, thank you. We like our hoodies. They're very comfortable. And if I should say, if you would like to get a hoodie, I would encourage you to use the platform and publish with it. And we may just send you a hoodie as a thank you for being a user. All right, well, I think we're 10 minutes over the hour. Thanks to everybody who stuck around. Again, we're happy to help you if you just want to reach out to us at engage@kbase.us or via the help board if you have any questions or interest in collaboration. Maybe you are putting together a proposal and you would like to integrate KBase into that proposal. We're happy to write letters of support and think with you all about how to utilize the platform. So thanks again, everybody. Again, my name is Benjamin Allen. You can reach out to me anytime and I will help you out. So thanks again and have a great rest of your day.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.